I'd like to echo Karen's thanks to the University of Illinois and to Susan and all the Children's Environmental Health Center investigators for hosting us here today. This is an awesome experience to get to know you and to formulate some collaborations. And I look forward to seeing many of the students that I met at the poster session at the Society of Toxicology meeting in New Orleans in March. Okay, so today my goal is to first provide you with a brief introduction to this term of epigenetics, which has been thrown around a little bit already today, and then to tell you 4 reasons that I think matter when you're looking at epigenetics as a mechanism that might mediate the developmental origins of disease. So we'll go into a little bit about dose, and I've been widely impressed here with the amount of doses that you guys are doing in your phthalate PPA exposure models. The second thing I'd like to highlight is diet matters, which also we see as a common goal here between our 2 centers. And then touch a little bit more upon what Dr. Peterson talked about, is that life course matters. So to really investigate how in vitro exposures affect health later in life, we have to follow either the animals or the human subjects across the life course. And then finally, at the end, I will just touch a little bit upon how future generations matter, which will really dovetail nicely with the data that you saw from Dr. Flaws this morning. And hopefully throughout the whole talk, you'll begin to see about how our conceptual framework at the University of Michigan and our Children's Center invades all of this work and looking at sort of the double hit, the nutrient-toxic interaction models that we've been talking about. Okay, so there was a preview. These are the Agouti sisters. So I'd like to begin by telling you a little story, a tale of 2 mice, or why DNA is not necessarily your destiny. So not surprisingly, these 2 mice don't look much alike, do they? One's brown, the other's yellow. The brown one's lean. The yellow one could stand to knock off a few ounces. But what might amaze some of you, although I think some of you know the punchline here, is that these 2 are not only sisters, but they're genetically identical. They're reared from the same mother with the same prenatal and postnatal environment, so what makes them look so different? So to understand that, we have to go back all the way to 1953, when the now familiar DNA double helix made its debut on the cover of Nature. So this pioneering work of Watson and Crick paved the way for modern genetics and the eventual Human Genome Project, which identified the approximately 20,000 genes in our genome that determine much of who we are, how we grow, or whether we remain healthy or get sick. But the Genome Project didn't tell the whole story. It failed to identify the instruction book that tells these genes when to turn on, where to turn on, and of interest to those of us here in the room, how to react to various environmental or nutritional influences. So all of that is the job of the epigenome. So I described this graph in great detail. We'd be here all day. So instead, I'd like to introduce a simple analogy, and that is one of the computer. So all of us probably have a computer, and on that computer you have one hard drive. So that represents your genome. But our computers have numerous different software programs. So that represents the various epigenetic profiles that represent the different cell, tissue types, and reactions to various environmental exposures. So here's how it works. The epigenome is comprised of a series of switches and markers. And the most extensively studied marker is DNA methylation, in which a quartet of atoms sits down on DNA and usually turns these genes off. So it's like a dimmer on your light switch. A gene that lacks methylation can be brightly expressed, but when that same gene gains DNA methylation, it can be more dimly expressed. So you have a variation here. A second epigenetic modification occurs on these histone protein tails, in which there's many modifications, including methylation, acetylation, ubiquitination. And these histones help compact the 3 meters of our DNA into cells that are micrometers in size. So this solves a packaging problem, but it also creates opportunities so that we have cells that are inactive for certain gene expression profiles, but they're poised for action. So getting back to these Agouti sisters, in the Agouti gene, in the yellow mouse, it is unmethylated. It's expressed all the time when it normally should be on only in hair follicles and only during one particular stage of development. In the genetically identical sisters, the brown pseudo-Agouti mice, this gene has a ton of methylation. So that shuts down what we call the ectopic expression of this gene, and it recapitulates a wild type brown agouti mouse. We know that the epigenome is vulnerable during multiple periods of development, but it is most vulnerable during the very early stages of development. And that's because there's two waves of reprogramming that occur at this time. So the first occurs in the primordial germ cells, where there's a complete erasure and remethylation event that erases any acquired epigenetic marks that occur in the previous generation, and then sets up these very sex-specific imprint marks that are really important for about 1% to 2% of the genome in mammals, and these genes happen to be most related to growth and development. So the second wave of epigenetic reprogramming occurs at fertilization, when there's an active demethylation of the father's genome, followed by a passive demethylation of the mother's genome. And you can see that certain regions of the genome are protected during this time, and this includes some, but not all, of the repetitive content, but also the imprinted genes that were set up in the previous generation. Then following this demethylation event, with all deference to Randy Jurgel, who was my advisor during my thesis at Duke University, and Michael Skinner, who are two of the sort of forefathers of this environmental epigenetics movement, this graphic should be numerous. These lines should be numerous in number, representing all of the different cell and tissue types that we have. Because this is what happens during differentiation. You establish different DNA methylation patterns. So one can imagine that any environmental or nutritional effect that occurs during this generation can set up an individual for a slightly different trajectory. So over a 70-year lifespan, this can lead to very consequential effects. And then any environmental exposure that happens during this reprogramming event is what we call has the potential for multi or transgenerational effects, in which you would look down through the grandchildren and the great-grandchildren generations. So this represents some of the conceptual paradigms that we're working on at the University of Michigan, and two of the chemicals that I'll talk about today. One, bisphenol A, has been well introduced, so I don't need to tell you a little bit much about that. But the other that I'm going to talk about today is lead, in which we know is, from many decades of research, is a potent neurotoxicant. But our center is especially interested in looking at whether lead also has an obesogenic effect because of some early animal work that followed male mice through 1 year of age, and also through some really interesting work that goes on at the University of Rochester looking at adipocyte differentiation. So we take a mouse to human approach, in which we use the agouti mice as a sort of initial indicator of whether the coat color shifts and whether our environmental exposures are affecting the epigenome. But then what we do is we follow their wild type litter mates, and we follow these guys out to about 10 months of age, so we can figure out whether the exposure has any effect on these life course trajectories. Also of interest in the animal model is we have access to our target tissue of interest. So now when you're dealing with epigenetics, it matters what tissue you're looking at. So one of the things that we're struggling in the field in our children's environmental health centers is we have an epigenetic working group. Can you use things like blood or fecal matter or other biologically available tissue specimens to represent your target tissues of interest? Does it look like the epigenetic profile in liver, brain, adipose tissue, et cetera? So we can model that in our mouse cohorts. And then we have the advantage to look at two prospectively collected birth cohorts that Dr. Peterson just introduced to you. So one of the first things I'd like to harp on today is that, and I think you guys know this, is that dose matters. So this is a coat color distribution of offspring whose mothers were exposed to bisphenol A put in the diet at the milligram level. So what we see is the statistically significant shift towards the yellow unmethylated coat color. So you have about 30% of the animals who are exposed to BPA showing this yellow coat color, which we know leads to obesity later in life. But interestingly, when Olivia Anderson redid, this was some initial work that I did at Duke in 2005 to 2006, when Olivia Anderson went to repeat this at the University of Michigan, we saw the exact same effect of the milligram diet, which was great. But when we dropped the dose to putting BPA in the diet at the microgram and nanogram level, what we saw was a shift towards the brown coat color phenotype. So this was some of our first evidence that epigenetic mechanisms following in utero BPA exposure may be non-monotonic. But we humans have an agouti gene, but it doesn't have this parasitic piece of DNA called a retrotransposon, which is why these mice get fat. So we were much interested in looking at a more genome-wide level at genes that are of more interest to humans. Can we also recapitulate this effect? And so what we did was use next-generation sequencing to query the entire epigenome. And what I'm showing you here is just the 1,500 base pairs that are upstream and downstream of the transcription start sites of genes. But we have data on the entire genome. Let's talk about high-dimensional data. And you're seeing here animals who are exposed to 0 BPA, our microgram dose, or our milligram dose. And if you stare at this data long enough, you can begin to see some trends. So here are a number of genes who are gaining methylation, but only in the high dose. Here are a number of genes that are gaining methylation in both the medium and the high dose. But in the middle, we have a set of genes that are changing their methylation only in the middle dose here. And you can see the same thing happening when you lose DNA methylation. So these represent new sets of genes that are acting in either a linear or a non-monotonic way following BPA. And so this was measured in DNA from the liver. And one thing that we really enjoy doing for this study, we weren't able to do comparative gene expression using RNA-seq. So instead, we took an approach and looked at the comparative toxicogenomics database. And you can see any of the genes over here already had evidence in the literature that their gene expression changed following BPA exposure. So we were able to now show that DNA methylation was changing at the same time. Moving to our lead exposure study, this was led by Chris Falk, who just started his faculty position at the University of Minnesota. We looked at the coat color again of animals who were exposed to different levels of lead put into the water. So this represents about 4 to 5 micrograms per deciliter. Then this goes up to 25 and 30 micrograms per deciliter. So remember, we are interested in life course exposures. So although we don't often see these blood lead levels in children anymore, we did in the 70s. And so those are the people who have grown up in the 70s and before. So this is at the level of the current CDC action level. And looking at the coat color shift in the agouti mice, we again can see this non-monotonic effect, where you're seeing an increase in the number of yellow unmethylated mice in the high exposure group, but an increase in the brown methylated group here in the low exposure group. So now our second exposure where we're seeing this non-monotonic effect. And Dr. Jerdal also has seen a similar effect looking at low dose radiation. So moving on to diet, this is one of those iconographics that Autumn Burnell and Randy Jerdal developed for this review article that they wrote in 2010 describing some of our results from our initial BPA study. So in 2007 at Duke University, we exposed the mothers to BPA in the diet, but we also gave them different background diets. So a background diet that was high in methyl donors like folic acid, betaine, vitamin B, B12, or a background diet that was high in genistein, the phytoestrogen found in soy. And what happened in this case is that the nutritional supplementation of the mother's diet negated the effect of BPA. So this was some of the first evidence that DNA is not necessarily your destiny. You might be able to alter your epigenetic profiles by different dietary and perhaps pharmaceutical approaches. There have been emerging evidence in the human literature that supports this hypothesis as well, including the Dutch winter famine, in which this happened in the winter of 1944 to 1945, in which there was a population that was severely nutritionally deprived. And I've been in talks where people argue about why this happened, but it is a combination of a Dutch railway worker strike and then a Nazi embargo on the food supply, so that women who were pregnant were eating about 500 calories during this winter. And what these investigators did is 60 years later, they recruited these individuals who were born or individuals who were conceived during the famine, and they took their blood and looked at DNA methylation at those imprinted genes. And this is an example where they saw a decrease in DNA methylation at the IGF-2 gene 60 years later, showing, and this was only present in individuals who were conceived during the famine and not born during the famine. So this points to that really early window of vulnerability as important for epigenetic modification. So you'll see that all of our animal studies start our exposures prenatally prior to mating, so that is on board in the mouse at day 4.5 is when the epigenome is reprogrammed. And then some elegant work by Rob Waterland has also shown effects of season of conception in Gambia and DNA methylation at these funky genes we call metastable epiallels, of which the agouti gene is one as well. So turning the tide a little bit, as part of our PO1 Children's Environmental Health Center, Lisa Markowitz, who is here, worked with, she is a registered dietitian herself, but she worked with other dietitians in our university to develop some really cool high-fat diets. So I didn't get a chance to ask you all at Illinois exactly what's in your high-fat diet, but we have made what we call humanized high-fat diets. And we have a Western high-fat diet and a Mediterranean high-fat diet. And this represents different ratios of the fatty acids that is in the Western diet or a traditional Grecian diet, but also changes the carbohydrate and the sugar content as well, and has some differences in micronutrients. And Lisa always has a great punchline in her talks, and she said if we were able to do exactly what we wanted, the animal protocol, people would not let us get the Western diet where it was so nutritionally deplete. So those profiles aren't exactly representative of what we're exposed to. So we are now running BPA on top of high-fat diet, and I'll come back to it. I'll talk a little bit about that in a minute. But this is some of our first preliminary data that was on her poster. So we're looking at 8-isoprostane, which is a long-term measure of lipid oxidation. So we're also profiling the dams in this study, which is the first time we've done this. So these dams, representing control plus BPA, Mediterranean plus BPA, Western plus BPA, were exposed to these diets for about 8 weeks. So 2 weeks prior to mating, the 3 weeks of mating, and 3 weeks of lactation. And we see no statistical difference in the isoprostane levels of the dams. But if you then move to the offspring, and this is our first time point of collection where we're looking at postnatal day 10, we're seeing a sex-specific effect of changes in isoprostane in the female offspring. So our next set of data will begin to look across the life course to see if this persists till 10 months of age. Okay, so it's a little bit out of order here, but now life course matters. And this is the design scheme of the BPA exposure study that I just showed you some data from. So we are doing the prenatal exposure and following throughout lactation. There's the 6 groups. I just showed you data from PMD10 and from the dams. These are represented on Carrie Nair and Lisa Markowitz's poster. And what we have left to analyze is our life course phenotypes. So now we take advantage of the Michigan Nutrition Obesity Research Center where we can do animal phenotyping, and we can look at lean and fat body mass. We can do CLAMS, which looks at energy expenditure and spontaneous activity, and we do this at multiple times throughout the life course. And at 10 months of age, the animals are sacrificed. There's also a fasting glucose tolerance test as well. So a lot of that data we're analyzing now. But I wanted to move back to our first BPA exposure study at Michigan, which Olivia Anderson led. And this had a very similar study design, but looked at BPA across different doses. So these are the mice that you saw with the non-monotonic effects on the liver. But here we're looking at the effects on horizontal and vertical activity. And you can see that immediately we noticed differences between males and females, so they're stratified here. But the most significant effect here is that in females, no matter what level of BPA they were exposed to, we're seeing increased activity, and this is present across the life course. So this was a few years ago when BPA was being touted as an obesogen on the cover of JAMA, and we were a little bit, what's going on here? Because our animals are hyperactive. And then we looked at our body composition, they were also lean. And so this is what really led us, doing this life course analysis like that, led us to go back and look at the literature and look at 2 things, the exposure timing and the underlying diet that people were using. And that's why we decided to repeat the study, looking at only the microgram dose with these high-fat diets that I've just told you about. The other thing I'll talk about that what life course matters, if you can, I give Lisa and Carrie, who's about to start our phthalate exposures, a whole lot of credit. This takes a lot of time to follow mice for 10 months, and they're in multiple cohorts, so it's not just 10 months. It takes about a year and a half. One thing that we did stumble upon, which was a very significant finding in this first BPA study, was the first statistically significant evidence that early exposure to BPA resulted in frank liver tumors later in life. So this was work by Karen Whitehouse. And so we can see that there's a linear increase in pre-neoplastic and neoplastic lesions across our dose levels. And then a non-monotonic increase in what we call multinucleated hepatocytes, which is on the pathway to liver cancer, only in the low dose. And so this recapitulated an NTP study from 1982, which used BPA doses that were 1,000 times higher than ours in adult animals, and so now we're seeing this in animals who are exposed in utero. So this was a serendipitous finding. We did not set out to become experts in liver carcinogenesis. Another interesting effect when we look at lead and we follow throughout the life course, we were able to identify that in utero exposure to lead leads to increased body weight, but only in the male offspring. So you can begin to see this across 10 months of age. We have these dips here in body weight that are represented on our arrows, and you may wonder what is happening there. These are when our animals were transported across campus to the phenotyping core, and they had this stress-induced reduction in body weight, which I really wish happened to me as well, but nonetheless, this really, I put this up here because you have to think a lot about when you're following cohorts of mice for 10 months of age, and now we keep all of our mice at the phenotyping core to avoid this stress-induced reduction of body weight from going back and forth across campus. Now, one thing that Dr. Peterson alluded to that we're very interested in figuring out is epigenetic drift across the life course. So this is relatively easy to do in our animal models. Our surrogate, so you have to have a tissue where you can get it more than once from an individual, right? So we're using tail. So you can get a snip of tail at 3 weeks of age, and then you can get a snips of tail later in life. And what we're looking at is whether the exposure here to lead enhances or moderates the rate of change. So these are some genes. This is a metastable epithelial similar to beauty, and these are free imprinted genes in mice. And so anything where you see an enhancement, this means that having lead on board increases the rate of change of the gene across time. So we saw an enhancement at CAB and age 19. We saw no effect at IGF-2, and we saw a moderation at IGF-2R. So this is just some beginning exploratory data, because even though we have the human epigenome roadmap now, which resulted in 25 papers published in February in Nature, they're only looking at 111 different tissues, but only at one time point, and they have no environmental characterizations on that. So this is the first attempt to do that. Karen introduced the element cohort, so we have the opportunity to look at drift in element as well, comparing cord blood DNA to venous blood that's collected 8 to 10 years later. This is just looking at cord blood and what we call line 1 DNA methylation. This is a repetitive piece of your DNA that you want to be methylated. So what you see is with maternal bone blood levels, there is a decrease in methylation at line 1, although you guys are looking like, oh, that line is just not very interesting. So I really encourage you to think about this when you're looking at environmental epigenetics studies. We're saying, is this really significant? Is it really meaningful? And the epigenetics working group is working on a review paper that shows just how small the differences in environmental changes on the epigenome are compared to outcome changes. If you were to look at cancer, line 1 would be decreased by a huge amount. So I would point you in the direction of Jackie's paper that was published in collaboration with Brisa Sanchez, who's part of our data modeling core, which really goes over some really sophisticated statistical approaches to looking at these data, including how to adjust for batch, whether you want to look at all of your methylation points together or you want to account for the fact that they may not be independent. So stay tuned for the review paper. And this is our first drift data that we have in humans. So these are the 79 individuals who we have cord blood for, and then later in life, and looking at 2 imprinted genes. And what you can see is that across both of these imprinted genes, there's a decrease in DNA methylation, although it is, again, not a huge effect size. What we don't have yet on these data is whether the environmental exposure moderates the effect of the change. I think this is my last slide. I'd just like to really thank Dr. Claus for looking at F1, F2, and F3, because we have a deficiency in the field of environmental epigenetics in that in order to show transgenerational effects, you have to go down to the F3. And so many of the studies only take a cursory or no look at F2. And there's a provocative hypothesis right now in the autism field that it might be the increase of pharmaceutical drugs that were prescribed to pregnant women in the 50s and 60s, and the increase in women smoking during World War II that is responsible for the increase in autism that we see today. So this is just a hypothesis. It's semi-controversial because there's no data, because very few people are looking at the F2 because it was saying, oh, you have to go down to the F3, which is true to show this is the first unexposed generation. But when you're exposing the mother, you're exposing the offspring and the germline of that offspring, which becomes the F2 generation. And that's a really powerful exposure. So we need more people to look at the things like Dr. Claus is looking at. And with that, I'd just like to, similar slide to Karen, thank the vast number of collaborators and trainees that are working on this. And I've underlined and highlighted the people who contributed specifically to data in this presentation. Thank you.